A laboratory in Rotterdam. Scientists analyzing DNA samples from 2,500 men. They're looking for behavioral markers, genetic variations that correlate with specific psychological traits. What they discover challenges everything psychology has assumed about personality development. Certain men carry distinct genetic signatures, not random variations, systematic patterns affecting neurotransmitter processing, stress response, and social behavior. Patterns that create consistent psychological profiles across completely different environments and cultures. Patterns that explain why some men think, act, and perceive reality fundamentally differently than others. This isn't pop science speculation. This is published behavioral genetics research from institutions like Oxford, Cambridge, Duke University, and the National Institutes of Health. Peer-reviewed studies documenting how specific gene variants create measurable differences in male psychology. What emerges from this research is remarkable. The genetic markers scientists are documenting correlate precisely with traits historically associated with warrior classes, and in modern context, with what's called independent masculine psychology, hypervigilance, strategic thinking, comfort with solitude, resistance to social conformity, all linked to genetic variations that persist from ancient selection pressures. This is the science that explains why certain psychological traits aren't choices, aren't damage, and can't be socialized away. They're expressions of inherited biology. Before we dive into the actual research, drop genetics in the comments if you've always felt fundamentally different from others, not better or worse, just wired differently. Let's see how many people recognize this pattern. The foundation. Why behavioral genetics matters. For decades, psychology operated on a blank slate assumption. Personality was entirely environmental. How you were raised determined who you became. Trauma explained dysfunction. Conditioning explained behavior. Genetics were dismissed as biological determinism or pseudoscience. Then the research became undeniable. Twin studies separated at birth showing identical personality traits despite different environments. Adoption studies revealing children resembling biological parents they never met. Genome-wide association studies identifying specific genetic variations correlating with measurable behavioral differences. The science is now clear. Personality is approximately 40–60% heritable depending on the trait. That's not environmental, that's genetic. Your DNA creates predispositions, baseline tendencies that express across different circumstances. Dr. Robert Plomin's research at King's College London tracked 13,000 twin pairs over 30 years. His conclusion: genetics accounts for about 50% of psychological differences between people. Environment accounts for the rest, but not in the ways we assumed. What this means: two children can experience identical environments and develop completely different personalities because their genetic baselines differ. One child's genetics create resilience, another's create sensitivity. Same environment, different outcomes. The genetics create the foundation upon which environment builds. For masculine psychology specifically, the genetic component is even stronger. Research published in Biological Psychiatry shows that traits like aggression, risk tolerance, independence, and stress resilience are 60–70% heritable in males. These aren't learned behaviors. These are genetic expressions modulated by environment. This matters because it reframes the entire conversation. If a man exhibits independence, strategic thinking, or social selectivity, these aren't necessarily defensive adaptations to trauma. They might be genetic predispositions expressing normally. Understanding this transforms how we interpret male behavior. Not everything is pathology requiring correction. Some traits are genetic variants requiring understanding. Genetic marker. 1. The MAOA variant, the documented warrior gene. The first and most researched genetic marker is a specific variant of the MAOA gene, monoamine oxidase A, which regulates neurotransmitter metabolism. This gene has multiple variants, but one specific version has been extensively studied for its behavioral effects. The low-activity MAOA variant is found on the X chromosome, meaning males, XY, only need one copy to express the trait, while females, XX, need two. This creates much higher expression frequency in males. Approximately 30–40% of men carry this variant compared to 5–10% of women. Dr. Avshalom Caspi's landmark 2002 study published in Science tracked 1,037 individuals from birth to age 26. The finding: men with low-activity MAOA showed significantly different behavioral patterns than those without it, but only when combined with environmental stress. Here's what the variant actually does. It creates lower baseline serotonin activity. by increasing the speed at which monoamine oxidase breaks down neurotransmitters. Lower serotonin doesn't mean depression. It means different emotional processing, different risk calculation, different threat response. Men with this variant demonstrate consistent measurable traits, enhanced aggression in response to provocation, but not unprovoked aggression. They don't start conflicts, but they respond to disrespect or boundary violations with disproportionate intensity. The genetics create a tripwire, not a bomb. Increased strategic risk-taking. They assess risks differently. What others perceive as dangerous, they perceive as calculated opportunity, not recklessness. Strategic evaluation with different risk tolerance, higher pain tolerance. Studies show measurably higher thresholds for pain perception and faster recovery from painful stimuli. The neurotransmitter differences affect pain processing. Reduced fear response in threatening situations. fMRI studies show decreased amygdala activation in response to threat cues. They notice threats but experience less fear. The genetics blunt the emotional intensity of danger. Enhanced performance under pressure, where most people's performance degrades under stress. MAOA variant carriers often perform better. The genetics create stress-enhanced rather than stress-impaired function. These are warrior traits. Traits that would have been highly advantageous in combat, dangerous environments, or high-stakes contexts. Modern research from Duke University shows these same traits express in modern men as high functional independence without social isolation, low susceptibility to peer pressure or social conformity, calm demeanor in situations others find stressful, strategic rather than reactive decision-making, strong defensive response to disrespect or boundary violations, comfort operating alone without social support. Sound familiar? These are the documented traits of what psychology now calls autonomous masculine psychology. What makes this variant particularly interesting is its distribution. It's found at varying frequencies across populations. Research published in Evolution and Human Behavior shows higher frequencies in populations with historical warrior cultures or violent selection pressures. Lower frequencies in populations with long agricultural stability. The genetics were selected for. Warriors with this variant survived combat more often, passed genetics to offspring. Over thousands of years, populations facing regular conflict accumulated higher frequencies. The variant isn't random. It's evolutionary response to environmental selection. The modern expression creates an interesting mismatch. The hypervigilance that kept warriors alive registers as anxiety in peaceful contexts. The strategic violence capability remains present but controlled. The independence that made solo operations possible becomes isolation in social environments. The genetics designed for warfare persist in peace. Dr. Kevin Beaver's research at Florida State University documented this mismatch. Men with MAOA variants report feeling different or mismatched to modern social expectations. They're not maladapted. They're adapted to environments that no longer exist. Drop MAOA if you've been told you're too intense or overreact to disrespect. The genetics might explain why boundary violations trigger disproportionate responses. The variant also shows what researchers call context-dependent expression, extremely controlled in normal circumstances, extremely responsive when threats appear. The ability to maintain calm baseline, then shift rapidly to high-performance mode when needed. This isn't emotional volatility, it's sophisticated threat response calibration. The genetics create individuals who can exist peacefully but respond decisively when circumstances require it. The switch isn't malfunction, it's strategic capability. Understanding this variant explains why some men can't just calm down or stop taking things personally. Their neurotransmitter processing creates different threat perception and different response thresholds. Telling them to change is asking them to override genetic programming. Genetic marker 2, the 5-HTLPR variant, sensitivity and environmental plasticity. The second critical genetic factor is the serotonin transporter gene variant called 5-HTLPR. This gene has short and long variants, with the short variant creating what researchers call differential susceptibility, heightened sensitivity to environmental input. Dr. Jay Belsky's research at the University of California introduced the orchid dandelion hypothesis. Dandelions, long variant, grow anywhere, resilient but limited ceiling. Orchids, short variant, die in poor conditions but flourish spectacularly in optimal environments. The genetics create sensitivity, not weakness. Men with the short 5-HTLPR variant show remarkable patterns. Enhanced environmental awareness. They notice subtle social cues, environmental changes, and contextual details others miss. Not paranoia. Heightened perceptual sensitivity. Deeper processing of experiences. They don't just experience events. They process them deeply, extracting meaning and pattern. This creates both vulnerability to negative experiences and capacity for profound growth from positive ones. Greater environmental impact. Research shows they're more affected by both positive and negative environments than long-variant carriers. Bad experiences affect them more deeply. Good experiences benefit them more profoundly. A 2010 study in psychological science tracked 300 men over 15 years. Finding: short-variant carriers in negative childhood environments developed worse mental health outcomes than long-variant carriers. But short-variant carriers in positive environments developed better outcomes, more empathy, deeper relationships, greater life satisfaction. The genetics don't create deficiency. They create plasticity, capacity for environment to shape development more profoundly. For masculine psychology, this explains several consistent patterns. Many men with autonomous psychology report difficult childhoods that profoundly shaped them. The genetic sensitivity meant negative experiences created deep impact. Those experiences shaped psychological development in ways that wouldn't have occurred with different genetics. But the same sensitivity creates extraordinary observational capability. The heightened awareness of environmental cues becomes strategic advantage. The deep processing becomes analytical capability. The sensitivity that created vulnerability also created capacity. The short variant also correlates with pause and check behavior. Tendency to pause before acting, assess before engaging, think before responding. This appears as social hesitation or awkwardness. Actually, it's enhanced processing, taking time to analyze data before committing to response. This pause and check pattern is core to strategic masculine psychology. The assessment before engagement, the thinking before acting, the observation before participation. What looks like hesitation is sophisticated environmental analysis. Research from Stony Brook University shows the variant also affects stress response patterns. Short variant carriers have stronger initial stress responses, but better long-term stress adaptation. They react more to acute stress, but adapt better to chronic stress. This creates individuals who are highly alert to threats, while functioning well in sustained, difficult circumstances. Modern environments present chronic low-level stress that this variant handles effectively, but acute social stress triggers intense response. The genetics optimized for occasional high threats in stable environments. Modern life inverts this, constant social stress with minimal physical threat. Genetic marker 3. The OXTR variant, selective social bonding. The third genetic component is the oxytocin receptor gene, OXTR. Oxytocin is the bonding hormone crucial for social connection and trust. Variations in the receptor gene affect how strongly individuals respond to social bonding signals. Certain OXTR variants create reduced social bonding drive without creating social dysfunction. Dr. Shelley Taylor's research at UCLA documented this. Some individuals have genetic variants that make them comfortable with significantly less social contact than population average. They're not antisocial, they're differently social. Men with specific OXTR variants show consistent patterns. Normal social capability. They can interact, communicate, form relationships. The mechanics of socializing are intact. Reduced social need. They don't require frequent social contact for emotional regulation. Solitude is genuinely comfortable, not tolerated. Quality over quantity in relationships. They form deep bonds with very few people while remaining distant from most. Selective rather than broad social networking. Slow trust formation. They take longer to trust, require more evidence, but once trust forms, it's remarkably stable. This genetic variation doesn't create dysfunction. It creates different social optimization. Instead of broad networks providing diffuse support, narrow networks provide deep support. Instead of constant contact providing regulation, solitude provides regulation. Research published in Proceedings of the National Academy of Sciences showed this variant was likely advantageous in warrior contexts. Warriors needed deep trust with few comrades but couldn't afford broad social vulnerability. They needed selectivity. They needed capacity to function alone for extended periods. The genetics that created effective warriors create modern men who seem antisocial but are actually differently social. They're optimized for depth over breadth, quality over quantity, selectivity over inclusivity. The reduced oxytocin response also explains tolerance for isolation others find unbearable. Most people need regular social contact for emotional regulation. Oxytocin release from bonding provides stress relief. People with OXTR variants regulate differently, through solitude, internal processes, non-social activities. This creates fundamental difference in social needs. Most people genuinely need regular contact. It's biological requirement. People with OXTR variants don't have the same requirement. Solitude is genuinely restorative rather than depleting. The variant also affects response to social rejection. Dr. Naomi Eisenberger's fMRI research at UCLA showed that social rejection activates physical pain centers in most people. OXTR variants create reduced pain response to rejection. Social exclusion is noticed but doesn't create the same distress. This explains why some men can walk away from relationships without apparent distress. They're not suppressing pain, they're experiencing less pain. The genetics reduce social pain sensitivity. What looks like coldness is different neurological response to social loss. Comment selective if you've been called cold for not needing constant social contact. The genetics might explain why you're comfortable with solitude others find lonely. Genetic marker 4, the DRD47R variant, strategic exploration. The fourth genetic factor is the dopamine receptor gene DRD4, specifically the 7-repeat variant DRD47R. This variant is associated with novelty-seeking, risk-taking, and exploration behaviors. Dr. Chua Sheng Chen's research at UC Irvine found this variant at higher frequencies in populations with migration histories, people who left stable communities to explore unknown territories, the genetics that drove human expansion across the planet. Men with DRD47R show consistent traits, high tolerance for uncertainty, comfortable in ambiguous situations that stress others, strategic novelty-seeking, not impulsive risk-taking but calculated exploration with exit strategies, boredom with routine, need for challenge and variation, stability feels suffocating, selective hemophilia, love of novelty in specific domains, stability in practical matters. This explains autonomous males' tendency toward unconventional paths. The genetic predisposition creates discomfort with conventional stability. Unconventional career choices aren't rebellion. They're genetic expression. The variant interacts with stress response genes to create what researchers call stress-seeking behavior. Not pathological stress addiction, but comfort in high uncertainty, high-stakes situations that overwhelm others. Research from the University of Michigan shows this combination was ideal for scouts, explorers, advanced warriors, roles requiring solo operation in unknown territory. Modern expression creates individuals who thrive in unstable industries, crisis situations, disruptive environments. Genetic marker 5, the NR3C1 variant, stress resilience. The fifth component is variation in cortisol regulation genes, particularly NR3C1, affecting glucocorticoid receptors. These variations create different baseline cortisol levels and stress responses. Specific NR3C1 variants create stress-resilient phenotype, higher baseline cortisol, constant mild alertness. What others call hypervigilance is genetic baseline, smaller spikes to acute stress, don't overreact to immediate threats, better long-term adaptation, handle chronic stress through biological adjustment rather than behavioral avoidance. This matches independent masculine psychology perfectly. Constant baseline vigilance, always scanning, always monitoring, not pathological anxiety, higher genetic set point for alertness. Dr. Bruce McEwen's research at Rockefeller University showed this variant also affects sleep patterns. Lighter sleep, more environmental awareness during sleep, faster wake response, advantageous for warriors needing instant threat response. Modern expression creates chronic sleep difficulties. Drop hypervigilant if you've been told you're too anxious or always on edge. The genetics might explain your constant environmental scanning. The integration, what this actually means. So that's the documented genetic evidence. MAOA variants affecting neurotransmitter processing and threat response. 5-HTTLPR creating environmental sensitivity and plasticity. OXTR, reducing social bonding drive and creating selective attachment. DRD47R, creating strategic novelty-seeking and exploration drive. NR3C1, affecting stress resilience and baseline vigilance. Together, these create a psychological profile that's remarkably consistent across cultures and environments. A profile characterized by strategic independence, selective social bonding, enhanced threat awareness, comfort with solitude, resistance to conformity, calculated risk-taking, performance under pressure. These are warrior traits, traits selected for over thousands of years in populations facing regular conflict, traits that persist in modern populations despite peaceful environments. The genetics aren't destiny. Environment still matters, development matters, choices matter, but genetics create foundation, baseline tendencies that persist across contexts. If you carry these variants, you're not broken, you're not damaged, you're genetically variant. Your traits served evolutionary purposes even if modern society doesn't always understand or value them. The hypervigilance was survival. The independence was operational necessity. The strategic thinking was combat advantage. The selective bonding was protective. The stress resilience was essential. Modern peaceful society is only 70, 80 generations old. Not enough time for significant genetic change. You're carrying ancient adaptations expressing in modern context. The mismatch creates friction, but the genetics are functional. If you know someone with these traits, understand. They're not choosing to be different. They're expressing genetic predispositions. Attempting to change them is fighting biology. Better to understand and work with genetic reality than pathologize difference. And if you're interested in human diversity, understand. Personality has substantial genetic component. These differences deserve understanding and acceptance, not pathologization. The warrior gene research explains why certain men are fundamentally different, not better, not worse, different, genetically different. Understanding the science transforms the conversation from "why can't you be normal" to "how do ancient genetics express in modern context?" That's the better question. Final question, drop warrior genetics if you recognize these patterns in yourself. Which trait expresses most strongly? Hypervigilance, independence, selectivity, or stress resilience? This is Primal Mind. Until next time, understand your biology.